I'm sorry. How do you?
Okay. That's one.
Okay, so good morning, everyone. My name is Gabrielle Alston. I'm currently a senior. And this summer, I had the honor and the privilege of working under the mentorship of Dr. Robb MacLellan with assistance of Dr. Nakamura and Dr. El-Nachef at the Center for Cardiovascular Biology at the University of Washington South Lake Union campus on a project entitled Enhancing Cardiomyocyte Proliferation to Improve Cardiac Cell Therapy. Oops. Oh, I'm sorry.
Hit the space bar on the keyboard.
Okay, thank you. Before I delve into the specifics of my research, please allow me to give you some background information to further your knowledge of my experiment. So heart failure is an end-stage chronic heart disease in which the heart becomes too stiff or too weak to effectively pump blood to the rest of the body. 50% of patients diagnosed with heart failure end up dying within the first 5 years of this diagnosis, and heart failure costs the U.S. about $30.7 billion annually. So the problem is that when a heart becomes injured, for example, by heart failure, the heart loses its ability to regenerate this dead heart muscle. So your heart cells, known as cardiomyocytes, which I will also refer to as CMs, lose their ability to proliferate or to make more of themselves after they leave their embryonic phase. So there is no treatment for heart failure, but the next best thing would be preventative measures, which would include medication to help improve your quality of life, restricted diet, and daily exercise. The next best thing to a treatment would be a heart transplant, but the problem with this is that out of the 1 million Americans with heart failure, there are only about 5,000 donor transplants that happen per year. And so the problems are that there are not enough donor hearts, and that the problems that come along with taking immunosuppressions may be too great for the patient. So the question that arises is, can we use stem cell therapy as an effective method to treat heart failure? So the idea would be to differentiate stem cells into cardiomyocytes and then deliver these newly made cardiomyocytes to the patient. And the diagram below shows a non-human primate heart that has been injected with newly made cardiomyocytes, which have engrafted, shown in green. So if you're wondering, what is a stem cell? A stem cell is an undifferentiated pluripotent cell. So essentially, a stem cell can differentiate to be a multitude of different cells. But for the purpose of my research, I focus on differentiating stem cells into cardiomyocytes, and I used a human stem cell line. So in order to get these cells to proliferate, make more of themselves, we have to look at the cell's epigenetics. And epigenetics are signals that tell a cell which gene to turn on. And it's been shown that an epigenetic enzyme, KDM4D, increases the amount of proliferation in heart cells. So in these images, courtesy of the MacLellan lab, shows a non-human primate heart that has been given an excess amount of KDM4D, shown to your right. And the results are that the actual size of these cells are not increasing, but actually the amount of cells once KDM4D has been introduced to these cells. So in order to introduce KDM4D, in my experiment, I used 3 AAV6 viruses, which are safe to use in humans. The first one was a KDM4D virus with a GFP tag, and GFP is basically just a fluorescent molecule, along with our control virus, also with this GFP tag, and lastly an M-cherry virus, which just stains anything that remotely relates to a cardiomyocyte red. Here's a list of my materials. And so the purpose of my experiment was to examine the effects of KDM4D expression on the CM proliferation. And we hypothesized that these AAV6 viruses will increase the amount of KDM4D expression, leading to this adult CM proliferation. This now brings me to our methodology, in which we first packaged our stem cells, differentiated these stem cells. We had the infection with the AAV6 viruses, the confirmation of gene delivery, and lastly, assessed cell proliferation. So first, for the packaging of stem cells, we picked our stem cell line, which again was a human stem cell line. Then we passaged them in 6-well plates, washed them with PBS, incubated them just to allow the cells to grow and make more of themselves. Next, for our differentiation timeline, we added ROCK inhibitor, chiron, and activin A before day 0, and day 0 essentially just means that this is when the cells will start to differentiate into cardiomyocytes. And after day 0, we added chiron, ZAB, and did multiple media changes. Next, we stained for troponin I, which is a protein only found in heart cells, so this would just let us know that these cells actually did become cardiomyocytes. Next, for virus infection, we removed the media, added 2 different concentrations of virus, which would include a high and a low concentration. We incubated them, brought up the volume, and this took about 2 to 4 days to see protein expression. This now brings me to my results section. This is actually my first attempt at differentiating my stem cells into cardiomyocytes, and as you can see, I did get beating cardiomyocytes. And next, this is when I stained for troponin I, seen in red, so this did tell us that these were cardiomyocytes. And we stained for DAPI, seen in blue, which just stains the DNA of these cells. Next, for our M-Cherry results, again, this just stained anything that remotely related to a cardiomyocyte. So we did see in both the low and the higher doses that we did get staining for these. Next, for our control for our KDM4D virus, we did see this GFP for fluorescence. And also for our KDM4D virus, we again did see this GFP fluorescence, and both of these were in the higher doses of the virus. So since we did see this GFP fluorescence under the microscope, we wanted to further confirm that the cells had been infected with these viruses, so we looked at the RNA of these cells. For the GFP RNA levels, we did see that both the control and the KDM4D viral infected cells did show GFP in their RNA, but it showed that our control virus had a higher viral infection than our KDM4D virus did. And then our KDM4D RNA results, well, again, we didn't expect to see KDM4D in the RNA of the cells that were infected with the control because the control virus did not have KDM4D in it, but the cells that were infected with this KDM4D virus did show KDM4D in their RNA, so this just let us know that the virus did work. This now brings me to my discussion. So to reiterate my hypothesis, we hypothesized that these AAV6 viruses will increase the amount of KDM4D expression leading to adult CM proliferation, and this hypothesis is pending. This is because we did differentiate the stem cells into cardiomyocytes, we did have infection with the virus, and we did see protein tag expression, but we weren't able to see this adult CM proliferation. So for future research, since KDM4D did have the lowest viral concentration, we may want to use higher doses and also optimize our infection protocol. And lastly, to further examine the critical role of KDM4D in this adult CM proliferation to become an effective treatment for heart failure. Thank you.